All right. So, good morning, everybody, and thank you for coming today. You can tell that our speaker today, Don Blancher, is an old chap, an old buddy of the Sea Lab, with all the shaking and hugging he's been doing over the past 5 minutes. And it is because Don was associated with the Sea Lab way before many of us were. After a PhD in environmental engineering from the University of Florida, he came to the Sea Lab in 1979 as a postdoc, a research associate, and then he joined the University of Alabama faculty as an assistant professor, where he was based out of the Sea Lab. That was the time when Tom Hopkins and William Schroeder were here as well. So he was one of 3 UA faculty here at the Sea Lab. That tells you a little, idea of how the Sea Lab has been evolving in terms of faculty representation from the different schools. And then in 1983, he went to South Alabama, to the main campus. He became an assistant professor at that school in the computer science department, doing a lot of computing work. And you will see that it shows when, in those slides with plenty of equations, you can tell that he has a lot of computing skills. Anyway, and then 2 years later, in 1985, he went to the consulting business, and he has had a very prolific career in consulting for over 30 years now. He has been doing a lot of environmental work in the Gulf of Mexico with a variety of things, impacts of all kinds of sorts and sources. And he has hired a number of former students here coming from the Sea Lab. So he has been very gracious and good to them, giving them an opportunity to learn new skills and a stepping stone for other jobs or as a permanent job. For example, I'm thinking of John Lerder, who did consulting for a few years and then he went to EPA. I'm thinking of Amy Hunter, who has been doing consulting ever since she graduated, and Rita Pizzi, and now Meg Goker right there. So, he'll be talking to us today about some work he's been doing for the Deepwater Horizon spill, consulting work with teaming up with many people, looking at the impacts of the oil on a variety of things. And also, I thought bringing him today to the Sea Lab would be a good thing for your students, so you can see another outlet, another venue when you're out there looking for jobs, the consulting business, and see what they do and whatnot. So you want to talk to Don after his presentation, he'll be around, and please feel free to come talk to us, and then Don will talk to you. Okay? Thank you, Don. Thank you for coming.
Well, thanks, Juice. Appreciate it. All the kudos. Yeah, I look out here and only see one person who was here when I was here. Maybe I won't mention his name, but he's sitting right there. Mike was here years ago, so we're still hanging on, I guess, still doing things. I appreciate the opportunity to come out here. Today I'm going to talk about some of the work that I did mostly for the state of Mississippi, and then we carried on and did the same work in the state of Alabama for the Deepwater Horizon. I worked for the Natural Resource Trustees from the beginning. Actually, I worked in 3 states to begin with, then focused mostly on Mississippi during most of the spill. We're in all the negotiations, and if we had gone to litigation on it instead, which would have been sometime around 2019 or 2020, they were estimating a couple of years ago, I would have probably been one of the testifying experts for the state of Mississippi. Part of the reasons we did this work was because the feds had their modeling folks in there. They had several different groups doing some modeling, mostly offshore, and we felt like we needed to look more at the nearshore environment and work on trying to figure out what the injuries were in Mississippi and then in Alabama. So what we did is take some existing models, modify those models. I had been familiar with these models for years and had worked with them in freshwater systems. They had been used in saltwater systems before, mainly Galveston Bay, but we took it, improved the model, and then moved forward with looking at trying to determine the injury. It was about a 3-year effort, spent a little over $2 million, so it wasn't a short task. We had a lot of people involved. The authors you see here, mostly from USM. We had a team at USM who are our advisors. Then when we did it in Alabama, we had several folks from ADCNR who helped us with some of the calibrations here, and Amy Hunter was working on it as well with some of the Alabama work. I'll describe those reports. They're all available in the administrative record. There's a series of reports, including peer review reports of the model and everything else that we had done during the period of time, so we'll go through some of that. I only have 285 slides, so I'm going to go through quick. No, I only have 65, so we still have to move fast. We're only hitting the top and peaks of some topics that we could spend hours on, really, in talking about how these things work. Talk a little bit about the history and the model status. The modifications that we did for the NERDA application, the baseline calibration from Mississippi and Alabama, how we drove the model, the exposure scenarios that we used, some of the data that we had there, the results, and a little bit of details, and then the injury estimate that we came up with for both Mississippi and Alabama. AQUATOX is a simulation model, i.e. it's a mechanistic or deterministic model, not an empirical model. It uses differential equations to represent state variables, and it's been used for a number of years by EPA in a variety of situations. The current version we had peer-reviewed in July of 2015 that was paid for by the Gulf of Mexico program, EPA Gulf of Mexico. We also had input from some of the folks over at the lab in Gulf Breeze. Just to give you an idea of where it started, it really started with Dick Park's work back when he was at Rensselaer many years ago, 74. I knew it as a graduate student in 75 to 79 and worked with it. In fact, we had taken when I was here at the Sea Lab, I worked as an IPA to the Corps of Engineers for ERDIC. It used to be called WES, but now it's called ERDIC, the Engineering Research and Development Center. And I worked on macrophyte algorithms on some of the reservoir models there, was the work that I did back in 1980 and 81. And we had teams of experts that would come in and talk about their modeling approaches, and Dick was one of those. And that's when I met Dick Park, was in 1980, working with old Fortran code. So it was originally for the Office of Pollution and Office of Pollution Prevention and Toxic Substances. So it started out as more of a toxics model. That's one of the reasons why it's very useful, is because it is good for both eutrophication as well as toxicity, particularly for organics. Doesn't work with metals, so don't get any ideas you can play with metals. It still needs some work on the metal side of things. But you can see here, I just put some of the history down so that you can look at it later. I applied it to the Cahaba River back in 2001, 2002 for a TMDL. I also applied it in a reservoir, drinking water reservoir in Florida a number of years ago to look at the interaction of nutrients and copper, which they use to keep the algae down in the in the reservoir. And of course, from 2012 to 2015, we worked for the Mississippi DEQ and worked with USM in developing 4 particular habitats to look at: the marsh edge, protected and exposed beach, oyster reef, and sandy and muddy open water. And then we took that model as we developed in Mississippi and applied it to Alabama using driving variables from Alabama. So we'll get into that in a little bit. The status is we've got a couple of real versions right now. There's AQUATOX 3.1, which is the Nearshore Marine Environment, which was publicly put out in late 2014. That's the model that was peer-reviewed. We have a Mississippi version that we use for toxicity. It's complete. The only difference is some PAH petitioning code that's inside of it that was proprietary at that point, i.e. we didn't want to have BP looking at it before the case was over, so we left it kind of behind the curtain, so to speak. But now, since the settlement has taken place, all of this should be available for anybody to look at, and will be available. We have the baseline calibrations that we did from Mississippi and Alabama and all those studies, and then we updated the driving variables, like I said, to look at Alabama. Some of the modifications as a report. Again, this is summarizing the report in a few slides. We put this together for the peer review committee so that they could look at it, look at the equations and everything that we used. One of the things we did is we added a couple of gills that we hadn't had before, particularly veliger and spat. These were missing from the earlier versions. We needed to add that for the oysters. We added some extra age classes of fishes so that we could put more state variables in there. And this is the number of compartments that you can have in running the model. Obviously, it doesn't fully describe a complete food web, so you have to make some generalizations, i.e. when you talk about putting something in like croaker or black drum, you're really talking about the same thing. As a generalized cyanid was what you'd use. And the same thing with a variety of different taxa is we use what we call, kind of sentinel species to act for a particular state variable so that you can get the model to work. But you can put together a pretty decent food web with that and come up with some realistic simulations, hopefully, as you'll see, particularly on a trophic level standpoint. One of the things that we added, and this had been in the works for some time about trying to add this, was modeling physical changes in habitats using fractal dimensions so that you could go to a marsh, and if you have and this is important if you're looking at restoration as well. If you have very little marsh edge, your fractal dimension goes down to about 1, and as the number of streams and marsh edge increases, your fractal dimension increases, and we can relate this as refuge. This can be used as refuge for certain species such as shrimp. If you take, for instance, the Grand Bay marshes nearby, the highest one we could find was this fractal dimension up here of about 1.52. But to give you an idea of what that does, we took it for Point of Pines. I took the Point of Pines simulation and changed the fractal dimension from 1.5 to 1.2. These are the actual simulations in Penaids. This is a difference graph which shows you that as you increase the fractal dimensions, you get better survival of shrimp. However, it actually get little declines in fish, mainly because they can't capture prey as readily, so their biomass decreases a little bit. Probably not statistically significant, but it does. You can see how this works a little bit. We also added factors in here for burrowing and reef complexity, and the complexity due to macrophytes with different equations. So you can modify all these, and these are great as what modelers call, pots, a potentiometer, so that you can change the K of the flow through a particular storage. And so it gives you better control in terms of calibrating it to real data. And that's the real trick, isn't it? Trying to get it to match what you've got in terms of real data. We have, oyster reef and restoration phase. Just to give you an idea, you can do a similar thing by changing the complexity in oyster reef. Here, it's helping out some of the brown shrimp, mud crabs, blue crabs increase a little bit, grass shrimp increase the most. And you can so basically, and when we say grass shrimp here in the reef, we actually use it's a misnomer. It's really the snapping shrimp and some of the smaller shrimp in there. It's not actually grass shrimp that we calibrated for in the reef, but it gives you an idea that you do have some control over looking at the populations and looking at the biomass concentrations there, and then controlling it with the model. Chlorophyll A, previously, the carbon to chlorophyll ratio was a single number. As we know, with different algal groups, that changes. Now you can actually specify the carbon to chlorophyll ratio for each individual taxa. This is also helpful in calibration, particularly if you've got good biomass excuse me, of the different algal taxa. Many cases you just have a chlorophyll, so it's kind of hard to know what the taxa changes are, but if you do have the information, it is useful to calibrate. The oyster model was something else that went in there. There was a recruitment. If you start out with a sac oyster, you have recruitment and spawning to the veliger stage, promotion to spat, promotion to seed, and then growth and promotion to sac oyster. The toxicant burdens passed along with size, class, proportion, and recruitment. And these are some of the triggers for size class change. If you want to get into some of the details, those are in the report, so I refer to that. And just like I said, I'm going to jump over these pretty quickly, but just know that that's in there. The other thing we added was allometric equations for invertebrates. Previously, allometric equations were only for fish. In freshwater systems, most of your invertebrates are small, so the size, I'm going to say size doesn't matter in that case, let's say. But anyway, in the allometric equations for invertebrates, we added this in because of the larger invertebrates that you get in the marine environment. It's basically using the same equation that you use for any heterotroph then, and it kind of normalized that and gives us a little better capability with the invertebrates. On baseline calibrations, this is one of the list of state variables that was used in Mississippi Sound. This is for soft bottom. And basically the way you drive the model is you add in your estimates for detritus, you have the organic toxicant driver, you get your base nutrients going in there, primarily phosphorus, ammonia, and nitrogen. That drives primary production, which then drives secondary and tertiary production, et cetera. So you do have immigration and of different groups in here. So you do have a loading that can go in that gives you an idea, gives you at least a baseline number for these things, so you're not starting out with 0. What I forgot to mention, that this is a box model. It's a one-dimensional box model. If you're familiar with hydrodynamic modeling, you usually think of single, second, 1, 2, and 3-dimensional models. This is a single dimension, but it takes a column of water, let's say, out in Mississippi Sound, and you're simulating. We were simulating in these about a 2-kilometer square grid. So this is what happens within a 2-kilometer square grid, and then you average it out, and everything is expressed in grams per meter square later. Another thing I failed to mention, it does track carbon. The model is based on carbon, but you can convert to organic material or energy if you prefer. I know most people wouldn't want to think of it in energy. I see Meg smiling because we could start that. I said before, we could convert this to energy if you want, but we won't do that. The biotic compartments have been modeled in Mississippi and Alabama, include these species. AQUATOX also has a library, not only for sites, which have base data for the site, that you may have characteristics such as detritus and some of the physical characteristics, but for each biological variable, of course, there's all the parameters that go along with it. One of the useful features of AQUATOX is that it does keep these libraries in that can be used for the future, and this is where you can really go into calibrating. These are the organisms that we calibrated for and got. It took a long time to search literature and try to get as much information as you could out of the literature to drive the model. To give you an idea, these are just one of the pages for blue crabs. You can see the amount of variables that need to be put into the thing. Includes, of course, a variety of, let's see if I turn this on. You could have low oxygen effects, salinity effects, change your allometric parameters, spawning parameters. I'll go back a minute. The animal data, of course, has half saturation feeding coefficients, et cetera, all your growth parameters and everything here, the ratio gametes to biomass, et cetera, and the appropriate citation that went along. In some cases, you'll find that we use professional judgment. We tried to get an organism that was of the same size and use that to calibrate it where we couldn't find anything in the literature. Excuse me. So basically we have some new AQUATOX studies. We have one that's for reef. It's calibrated for different oyster reefs in Mississippi and Alabama. We actually have about 5 different calibrations that we used. Most of the driving variables here were for different salinities. And you do get differences because of those salinities, and it does end up with a slightly different food web calibration. We have marsh edge habitats with and without SAVs. We have soft bottom habitat, and then we have a variety of beach habitats simulated for 3 what we call wave energy spectra. We have the exposed shoreline, the high energy beach, back barrier, moderate energy beaches, and then protected shoreline like you see along Mississippi and along some of the coast in Alabama. That's more of the mainland type beaches. With these, we also have the capabilities to either have with or without SAVs. We have some instances where, for instance, back barrier islands, we've got SAVs, and in protected beaches, we have SAVs. So we do have it with SAVs as well. The process that you use for each model, let's say we take the marsh edge, you find your salinity, temperature, driving variables, and of course when you're talking about toxicity, you'll have the exposure variables for that particular location. get the support data for hydrography, any temporal information, that is time series of everything that you can get, including the biota. That's always the hard one, trying to get good time series for the period of time that you're talking about for the biota. You'll see that in a minute. Many times we only have seasonal data or maybe even annual data in many cases. So then we'll go a little bit into how we broke out the toxicity and the oiling as we get into it. We had to do some interesting things there. Typically you would have concentrations of these things and you could just drive them in. However, we didn't have continual concentrations that were available and the sampling wasn't insufficient. This gives you an idea of how we broke down the salinity regimes that corresponded mainly to the NOAA salinity regimes that we had, and kind of gives you an idea. This gives you an idea of the effect on salinity. This is you specify for each species, the respiration parameters, the mortality parameters, as well as the ingestion parameters, kind of like the old, environmental physiology stuff where you have the capacity adaptations and resistance adaptations. That is, the capacity adaptation is when an animal can thrive and reproduce and do everything else. And then there's a bound of parameters, whether it's salinity or temperature, where the animal can survive, but it probably won't reproduce and all. So you can specify these for each animal, and this is how you can get, by driving mainly salinity and temperature, get the food web to change. So you can start with the same food web and then run it for a few years, and you find out that certain things drop out. So just an idea of some of the time series for variables. We looked at both deriving temperature as well as we had some measured temperature. Then we'd go back to the any database we could find. This is a STORET database or existing data that EPA has for Mississippi Sound. And then, of course, we do have some measures of TSS. We took those and then we would derive variables such as Secchi disk. You can see some of the Secchi disk measurements versus what the simulation is right here. We did the same thing with MODIS data for chlorophyll. I can't read. I think this goes from like 2006 to 2014 in this particular parameter. We also had some NGI data over a period of time. We calibrated to the NGI data, which was actually done using liquid chromatography rather than the satellite data. You notice the satellite data typically overestimates chlorophyll a little bit, but we did catch some of the peaks and all, and this is one of the simulations that we did for, I think it was in Mississippi Sound. So, biological baseline variables, this is where the data starts getting even more sparse. Zoobenthos, and particularly at different sites. This was soft bottom in Mississippi Sound. We had some data for zooplankton for a couple of batches right here. And then you can look at, I think this is a Karsia down here, and then these are average biomasses for fish. Compiling this data and putting it in is probably one of the hardest tasks in trying to get enough information. And again, trying to get something more than just seasonal data is often difficult in these situations. So we try to utilize as much available data. I know Ken Heck provided a good bit of information that we were looking at. We got some of the information from the lab here, from some of the students that were working here at the time. So these are some of the locations that we use mainly for the Mississippi. The orange dots are locations where we took MODIS data. You can see the focal data set that we use. We even went back to the old Coastal Area Board data set, which Mike and I were talking about previously, that I was involved with in 1981 and 82 here at the Sea Lab. So we went back and tried to gather as much information as possible to put in here. When you look at some of the information, this is the past Marianne Reef. We took DMR data. They only collect it during a particular season, and so it makes it a little bit hard to know how well you're calibrating these things. But you can see that we had the data for seed, sac, and spat that was taken by DMR. You can see what the range is that they collect in the variance, and typically we tried to make sure that we could get the peak to correspond to the peak that we had in timing, as well as kind of the range. The best way of doing it when you start looking at this type of data where you don't have good continuous information is to just make sure that your mean and variance fall in. The goal of this was to get a decent calibration for total productivity of the system in these different habitats, and then look and see how the oil impacted that productivity. So in this particular case, we can look at the seed and sac oysters. What this means is that the relative bias indicates that the mean is the same within a 95% confidence interval right here. And then the F statistic over here tells you whether or not the variance is the same. So as long as you fall within this ellipsoid right here, you know you're dealing with a population that has the same mean and variance. So it's often used by modelers, particularly in looking at biological variables where you have very sparse data and trying to see whether or not you're coming close to what the population would really be doing. So the food web setup and verification is another way of looking at it. This is the AQUATOX gives you an actual trophic level. We didn't look, one of the ones, we looked at this to see, you know, does this make sense? Do these animals, the trophic values that come out for these animals make sense? One of the ones that kind of bothered us a little bit was the blue crab. And you notice right here, it's running between 3 and almost 3.7 in terms of trophic level. You know, we assumed it to be a little bit lower down. The star represents some information that we got on stable isotope data that indicates that, well, according to the stable isotope data, we're probably right in between. It'd be great to go back here and look at all the different species and then calibrate for stable isotopes, and it would give you an idea whether or not that your food web is operating properly. We did it by having a series of expert scientists looking at it and saying, well, it makes sense, and looking at the, you know, particular, you know, if you're looking at some of the invertebrates, we had Chet give us feedback on that, Reed Hendon gave us feedback on the fishes, and Robert Leaf with menhaden in particular, some of the fisheries important species. And so, you know, they looked at a lot of the calibrations and said it's probably pretty good in terms of where things should be. This is the Cedar Point baseline productivity for the different species. You can see the constituents, mud crab and brown shrimp and sack oysters make up the large portion of the biomass, largest portion. And if you look at the overall number, 600 grams, when we negotiated productivity with BP and with NOAA, we ended up with a number of 580. We ended up a little bit higher productivity in some of the Mississippi reefs, but the 600 was based on a lot of the data that we got from around Dauphin Island and everything. That 580 was data that we had gotten to see what the total productivity of reefs are. If you take some of Peterson's work that was done on the East Coast, you'll find the productivity for oyster reefs get up to about 1,800 grams per meter square. And this is secondary production only. And so we felt like, you know, we're pretty much where we needed to be with total productivity. And so then we took that and that's where we applied the toxicity side. This gives you the baseline productivity for the different communities, just to give you an idea. The soft bottom, this has the fish on top of it as well. Soft bottom in this is, I think this is the Alabama one. I think the if you look at soft bottom productivity, you're looking around 150 for secondary, and then when you add fish on top, there's a good bit of fish in the water column there. Then the reef, you can see obviously that 600 had the fish in there because here's the 600 right here. A little bit surprising with some of the lower values here, but it's reasonable if you look at Peterson's range. All these fall within the range that Peterson put together and he compiled for NOAA in looking at some of these baseline productivities. From that, what we do is we applied the exposure to oil. I'll go through that a little bit quickly. The conceptual model was that the oil came inshore on the surface. It hit the shoreline, combined with detritus on organic materials and sediments and organisms, was processed into the system, and then slowly migrated back out offshore. This is the classic concept of oil coming to the shoreline. It's what NOAA used in their conceptual model, and we use the same conceptual model. Gives you an idea of the relative magnitude of TPAH from the NOAA NERDA data extractions that we had in Diver, which are all available now, and there's some huge data sets that you can look. If you're trying to look at specific locations, it's a little tough, but if you're looking at it generally, it gives you a pretty good idea. This is the difference between Alabama and Mississippi. We had some much higher concentrations in Alabama than we had in Mississippi. We did throw out a series of samples and water samples that were considered to be oil samples, that is, where somebody had sampled a floating slick and you can get really high numbers. So those weren't included in our analyses. We kicked those out. I think that gives us a very conservative approach in how we viewed the exposure. I argued with NOAA a good bit on this as to whether or not we should throw some of those data out. But in the end, we agreed to use the conservative approach because if we had to go into court with it, we didn't want having our data dinged, at least on that point, that we even used the wrong oiling data to begin with. So if you look at it, what we did is we developed a maximum likelihood estimate. Whether or not you're using, if you look at the detected concentration, the averages you get depend on the detection limit that you use. You can have a non-detect equals 0, the non-detect equal the detection limit, that is the method detection limit, and to calculate it. And you can see it generally follows kind of a log normal plot. So we decided to use a maximum likelihood estimate for both the water and the sediment data. And then what we did is we then fragmented that information from each state and for each location as to habitat. This is the water column data. I can't remember if this is Mississippi or Alabama, but you can see that the exposed beach, for instance, got the oil a little bit earlier. And this is taking the actual data. And what we did is we took the maximum likelihood estimate and then took satellite information in terms of where the oil was. You can go into IRMA, which is the NOAA site, and they binned where the oil was, what percentage of a 2-kilometer area was covered with oil, whether it was 25% covered or 50% covered or 75% covered. From that you can calculate a volume of oil and a mass of oil. You can take the depth and then get a nominal concentration from that. We took that exposure from the satellite information for a particular location and then normalize it using the maximum likelihood estimate to populate it with the actual data that we saw within that area. I don't know if that made any sense, but we can talk about that more. Basically, we used, Rouhani was a statistical expert that NOAA was using. We used his exports. And then it gives you the most likely distribution of the data that you'll have that kind of follows along. So you have a timing that was predicted by satellite and a concentration that's predicted by the log normal distribution. This is the Marsh 2011 data, which was highly log normal and fit the model very well. It's probably the best data set we have. When you look at all the individual data sets, you'll notice data sets like this, which was, I can't read it, well, I can't remember. It's probably one of the water column for 2012 or something like that. And so you see you have very sparse data, and our predicted level is this line right here. So you can see that when you have very sparse data, it starts to fall apart. But that's just a fact of where we were. We continued to use our maximum likelihood estimate. Now, the sediment exposures also were not uniform, so we decided, you could look at one spot and find where it was heavily oiled, and then you go over a few meters and you find there was no oiling. So what we decided to do is to use a percentile approach to most accurately represent exposure. So we took our maximum likelihood estimates and then took the 95th percentile exposure and then ran the model with that 95th percentile or the 99th percentile or the 85th percentile. So that meant for every model run, we would take a different exposure scenario to look if you were in an area that had the 95th percentile oil exposure that was observed, this is what the toxicity would be. So then, if that makes sense. So you end up with a tremendous series of runs, and this just gives you an idea for the, I think this is Alabama's data. It gives you an idea of the percentile approach. This is these are the concentrations of oil that, for instance, in the soft bottom, the highest it got to was a little over 1,000 parts per billion. If you look at the protected beach, it got up to 14,000 parts per billion. So this has to be the Alabama data. And that was the 95th percentile. Then we looked at that exposure, and you get down to a point where you're at 100 percentile. So we have habitats that we've calibrated with these different exposure levels, and now we know what the area of that habitat is, so we can then take that percentile and apply it to that habitat. So if you've got, for instance, the soft bottom, the 99th percentile, only 1% of that habitat was actually exposed at that level. And then we went down to 75 percentile because once you get down below 100 parts per billion, you really don't see any toxic effect. So there's no use in just following that curve on out. So most of the habitat did not see impacts. And how that translates, well, a little bit about pH models compartment. We also broke down the PAHs into bins, and we have representative PAHs that represented in each bin. This graphic right here shows you, for instance, the different PAHs, and then we sum them up to get to a total PAH, because what's reported in the database are total PAHs and the individual constituents. And so we had to take those bins and then calibrate it to the actual data that we had there. So glossing over it pretty quickly here, but it gives you an idea of the percent of TPHs in Alabama surface sediments. A lot of them were real heavy, and we just called it benzoate pyrene. It doesn't mean it was 45% benzoate pyrene. That's the benzoate pyrene bin, which included all your really heavy PAHs. So anyway, you'll notice that the oil that we had also was a highly weathered oil compared to the source oil. So we did have a lot of the higher level PAHs all here, and that's what we used to expose the organisms to. This gives you an idea of the an exposure scenario for Mississippi soft bottom. You can see the water column exposure comes in real quickly, and by August of 2010, it's gone. The sediments then peak up and then slowly degrade over time as it both dilutes and degrades and gets more weathered. And so you'll see that the concentrations go out all the way to about 2012. We played around a good bit with this in terms of looking at this lag curve to make sure that we got fairly decent agreement with the data as it existed out there. Bioaccumulation in AQUATOX, this is standard AQUATOX stuff, but to give you an idea is that you have uptake through the gill, you have uptake through food sources. The toxic food sources comes from a variety of petitioning and consumption approaches here. So you have the uptake that's coming from the diet, then you have the loss of toxicants due to a variety of factors, predation, mortality, depuration, spawning, etc. And then, and this is just kind of classic AQUATOX. If you're interested in looking at some of the bioaccumulation parameters, you can see the top level here is looking at how the log of the bioconcentration factor changes for fish. And you see that in AQUATOX we're using this line right here. It starts to drop off as you get to fish because the higher PAHs tend to pass through the gut a little bit more. It works a little bit better for the invertebrates. We stuck with and of course you use a different BCF for the invertebrates and the fish, but it gives you an idea of what's going on. Some other parameters here show you some different literature values of parameters and how AQUATOX compared. And then the metabolism rate, there is some indication in the literature that the metabolism rate, increases the elimination, but when you look at the actual data that's there, it's kind of hard to see that trend in terms of looking at that kind of scatter curve that you have there. Anyway, the way that you really go back and see how closely and how well you're doing in terms of that is to actually get some data where you've got data in the tissues of the organism. How am I doing? Oh, good time. Okay, not too bad. And we use the method of Vianney and used biotransformation from the Arnaud databases. We can give the literature is listed in the reports, and you can pull those things up. These are kind of the state of the art, expressions of bioaccumulation. We're presenting this information at CTAC in Europe in May at the bioaccumulation conference. The Europeans have been very big on this for a long time in terms of looking at particularly PAH bioaccumulation. So we're going to present some of this information here. This is a log-based graph right here. The data are from a variety of sources. We have the NOAA data, or the orange. In Mississippi, Shia had collected information for the health department, and then Milroy had collected some samples here. So it's kind of hard to see whether or not we come close to that. This is the lines are the different for seed, sac, and oyster, the 95th percentile. Obviously, some of these organisms were not exposed to the 95th percentile, but it gives us an idea of where we are in terms of range, that at least we're falling in the range there. We did not see any toxicity from this with the oysters, by the way. The toxicity, I'll explain a little bit of that in a minute. We also took any laboratory data that we had and tried to simulate what was going on. Vignier from, I think, Gulf Coast University was one of the NOAA investigators. We took his data where he looked at bioaccumulation in the veligers, oyster veligers, and looked at larval tissue. The graphic over here shows you the difference between the Vignier data and how AQUATOX treated that. We underestimate a little bit in phenanthrene and fluoranthrene and pyrene. We overestimate in chrysene. And when you look at the sum, we overall underestimate what the bioaccumulation was based on that. So we could certainly tweak it a little bit. We thought about tweaking it, but since we were already pretty high on chrysene, which is one of, it's a pretty toxic substance, so we decided that we would just leave it as is and run it this way. It's maybe a little bit conservative for some of the PAHs, but it does show us that at least we're in the ballpark and we're not totally crazy. You play with this long enough, you start believing your models, right? So, AQUATOX toxicity calculation. The way AQUATOX works, it really uses de Toro's toxicity approach, which is you reach a critical body burden. That is, you uptake toxicants until you reach a critical body burden, and the organism dies. It can also be used in terms of increasing respiration and some other things, so that you actually see what you're actually tracking is biomass of a population. So what you're looking at is some of its mortality, but some of it could also be a chronic effect, that is impact to feeding as well. And bioaccumulation and toxicity is a function of KOW, and so each time step that the model goes through then kills a certain percentage of the organism based on its internal body concentration, and that's summed up and accumulated for looking at the total kill. LC50 has a very strong relationship with KOW. The information that we used for data, NOAA had a fairly large toxicity testing program going on. We used some of the information that they had, and also BP had a toxicity program going on, and if it was published, we utilized that information as well. In DONAX, I think we did a little better job. This is the 75th and 95th% exposure. The green and blue lines represent what tissue concentration was in DONAX. Again, even with this, we did not see significant mortality in DONAX, even though it shows that the bioaccumulator is a pretty tough organism according to the LC50s that are out there. The yellow lines were taken, now the simulation is for the offshore Barrier Islands in Mississippi, so they got a little bit higher exposure, but not as high as what you saw in Alabama. And we took the NOAA Alabama, Florida samples. Those samples were only taken way out here in 2011, so we don't have any data in between. The unfortunate part is we don't have any data when the highest levels of bioaccumulation would have taken effect, which would have been in June and July of 2010. So what it meant is that we didn't get out there fast enough to really get a good idea of what the bioaccumulation was. So I'm trying to get some data from Louisiana. I know Bill Stickle had gone out and was collecting data in the Louisiana marshes using some polyethylene membrane devices which act like biota in terms of picking it up. You can see some that was done by the National Park Service on Horn Island during the late summer. And these are the concentrations of PAHs, which again are on Horn Island. So we kind of, the line passes right through the Horn Island data, is a little bit higher overall, but if you look at it, you can see that it tails off pretty quickly in 2011. With the 95 percentile, we see a little bit higher peak that comes back here, and it looks like out in 2011 in some of the NOAA data that they did have some peaks. It wasn't just a baseline. So we feel like it's we're probably simulating as best we could. It would be great if we had some data here to see whether or not our peaks are there, but it does give us an indication that at least we're on the right track in terms of tracking bioaccumulation. So how do we take all that and then put it into injury estimates? These are the areas that we assessed in Mississippi and in Alabama. We didn't go up in the northern part of the bay because there was no indication of exposure further up north in the northern part of the bay. We have the slightly different habitat categories. we have the spatial categorization of each of these, and then we applied the percentile approach to each of these things. These are some of the simulated areas that we had, mostly the yellow dots here in terms of where we simulated and where we have simulations for. We never got to doing the snapper reef. That was one of the ones we wanted to get to, but we really didn't have enough data or enough time. Had we not settled last May and got on a fast track to finish up, we may have had time to go out and do some of that, but that's where we stood. This gives you an idea of a difference graph of the 85th percentile soft bottom in Mobile Bay. You can see a little bit, what happens here, most of the kill when the oil spill came through in the water column was, of course, to things like acarcia and rotifers. And then we have a little bit of a peak that's kind of it's probably a response to, it's probably more of a food chain, depression right here. Same thing here as you get out into 2012. What you're looking at is this is not a toxicity effect. One of the nice things about AQUATOX is you can go in and see where the mortality is coming from. You can see whether it's predation or whether it's actually toxicity in there, and you can differentiate those. If you look out here, you see that there was a decrease in shrimp, but that's mainly because of the increases in some of the other organisms, including fish, in this group here, that probably caused a decrease in biomass due to predation. So it's not really you're not looking at toxic effects out here. What you're looking is at food chain repercussions that occur, and you see similar type things when you have any food web model. If you're familiar with AQUATOX, EcoPath. I took that first off and kind of whacked the model for a couple of days with some oil, and you see those gyrations occur for several years later in the food chain. Some go up, some go down. So I think from that perspective, you can look at these and dig into these and see whether or not whether it makes sense. But what we're trying to get to is what is the overall loss? What is the overall loss in terms of oyster reefs? In 2010, our simulation predicts that in the highest exposed areas where you had oyster reef, that is our 95th, 97.5, and 99th percentile. That's pretty high levels of exposure. We lost somewhere around 100 grams per meter square. So what you're looking at is each of these are the percentiles for 2010 for the entire year. In 2012, you started getting some recovery. In the highest exposed, the 99th percentile, we still would have expected to see some toxicity. And this is overall biomass. Again, remember, we're looking at about a level of about 600 grams per meter square. So it's looking at all organism. When we say oyster reef, we don't necessarily just mean oysters. In fact, if you dig out the oysters, you find out that particularly sac oysters had no effect at all at these levels. If you look at the production loss over grams per meter square over 3 years, the reef in Alabama did have the highest impacts, whereas in Mississippi, the higher impacts were in the marsh edge. And so, if you look at the overall per meter square, you can see this is accumulating all the percent losses based on all of those percentiles. So I've got a big pivot table that has all this data in it that comes out. Each run, for each location was all compiled together, and then we pulled all the information, threw it into the spreadsheet, and then sucked it out with a big pivot table. If you look at the percentage overall and percentage losses for each of the habitats, this is in Alabama, you can see that in the protected, the thing is we had higher losses in the oyster reefs, but from a percent of productivity, it meant more in the near edge environment when you look at this, particularly in some of the protected beaches. But when you look at the absolute value of what we're predicting, we're only talking about less than 5% of the productivity was impacted in Alabama at each one of these different habitats. And in the soft bottom, you see it's even less than 0.5%. And Ken, I don't know how much sampling it would take to look at a 5% change, but you would not see this type of change in any kind of sampling program. Typically, you're on the order of maybe you can detect a 10 or 20% change in a population with most sampling protocols. So this would have been below detection from anything that you could have done with a sampling program, I think, unless somebody had really intensively sampled those areas. So The reports are available in the administrative record. I can give you links. I sent the links to Juice, but I can send links to anybody. I'll give you a card if you're interested. And also, when you go to the administrative record, you can look at the peer review reports. You can look at all the reports are out there. We have some summary reports summarizing this information that we presented to the trustees. And of course, once you get in the administrative record, you can look at everybody's reports and data and stuff that's out there. How many records are in there, Meg? 15,000 records or something like that in the administrative record? So you got a little bit of data mining to do over the next few months anyway if you want to try to cover all that. So I'll stop there, take any questions, and hopefully it gave you enough insight into how we estimated those losses. The big thing was, both in Mississippi and Alabama, is that the NOAA studies did not really show any loss coming into the shoreline, which was really unacceptable to the Alabama Mississippi trustees, saying that there must have been something. How can we detect this? How can we at least demonstrate that we had some injury that occurred to our natural resources? So our goal was to see if we could measure that using the model, and that's hopefully what we were able to demonstrate. Again, if you tried to go out there and measure it, you wouldn't have seen it. And in fact, if even offshore, it was hard to measure and see the actual impacts. They do have some studies offshore that do show impacts, but then as you got closer to the source and you got closer to the source oil that was coming up, you had more toxic effects. So anyway, it gives you a kind of a generalized approach of how we did. Summarizes 3 years of information and a bunch of reports into an hour. So good, I did it. Thanks. Is everybody stunned? Questions? Go ahead.
Did you include fisheries harvest of oysters or fish or other species in your frequent models?
Yeah, we had Robert Leaf go through and build menhaden. So if you want to look at menhaden, Robert Leaf did a pile of menhaden data for us. And then we used the DMR data in Mississippi for all the harvest information. They go out and do quadrats. We had that data for several years. And the graphs where I showed the sac oysters and the seed oysters, that was DMR data. And so you got a pretty decent range and a good idea. One of the things that we were doing early on was that some of the assumptions were that, well, oysters are very, very productive. And they were looking at literature and getting up to 1,800 grams per meter square. And we don't think so. Not from the data we were looking at. We knew it didn't get there. In fact, in Mississippi, we were looking at, on average, about 4 adult oysters per square meter, which is actually a relatively low density. We actually had higher densities in Alabama. I think that's why we may have seen more impacts in Alabama, because your reefs are shallower, so they got exposed to higher sediments compared to the deeper reefs in Mississippi. In Mississippi, we had very little impact actually from a toxicity standpoint of the oysters.
So I understand this is all based on